Okay. Thank you, please. Okay, morning, please. Once again, we apologize for the technical hiccup. I think you can inform. I'm thinking the class will be informing the people that we've changed the. I don't know what to say. Is the link or what? But then, please keep the comments coming and indicate your presence. And when we had a sizable number, then we can kick off immediately. This is something that you need to be available, and you need to take down notes. Please don't rely so much on the videos you may follow later on. There is a teaching and a learning in writing. Okay, when you're listening and writing down things, and when you listening and writing down things, it's educative. Okay, you commit it to memory better. Okay. Yeah, it will be good for you to understand these things at first phase, and rather than wait for some other times coming. Okay. So last week or on Tuesday, we were able to introduce concepts in blood, and we spoke about the composition of blood, the properties of blood, the functions of blood. We also spoke about the measurement of blood volume. We also spoke about the regulation of blood volume. And then today we want to commence with an important subject matter, and that is plasma proteins. Okay, that is plasma protein. So that is what we want to talk about for the next couple of minutes. It's quite an interesting subject area, and so please try to follow keenly. And before we also proceed, we're trying to see how to be able to route the challenges that many of you have indicated that you're failing to upload or submit the assignment or the assignment I gave. Okay, walk around it. Walk around it, and we'll see how to do that. Okay. Okay. So far, so good. Once again, good morning, and hope you're keeping safe. Hope you're also trying to protect yourself. Okay. In as much as whatever that be, we need to stay protected. Okay. As medical students in training, you should also be a voice in your local community and inform them about and then so that you help enforce the regulations the government has given with respect to protecting the nation from this COVID-19. Okay, so last week or on Tuesday, beg your pardon, we spoke about blood, and we said blood is not only the corpuscular component and the fluid component, right? So today we'll look at the fluid component. I'm seeing somebody is mentioning that they are now light skin for continuously wearing mask. Of course, we'll talk about that some other times. But of course, also try to find out you put on mask, reducing your partial pressure of oxygen. So which is plasma, and we said plasma has about 90%, it's made of 90% water and 10% solids. And we further mentioned that within the 10% solids we have, it's made up of inorganic substances as well as what organic substances. And we went forward to talk about those inorganic and organic. Okay. And so today we also mentioned within the organic we have plasma proteins, right? So today we'll look at plasma proteins, and it's a mixture of simple proteins, glycoproteins or lipoproteins, and other forms of conjugated proteins and are present in blood. Okay, but then we can majorly group or classify plasma proteins into three. So we have albumin, we have globulins, and we have fibrinogen. Okay, so we have albumin, we have globulins. Take note, and we have what fibrinogen. Okay, and the average or the average amount of plasma protein is 7 grams per percent or 7 grams per deciliter of blood. So that means for every 100 mils of blood, you have an amount of plasma proteins worth 7 grams. Okay, and albumin is the most abundant plasma protein, and fibrinogen is the least abundant plasma protein. Okay, and then in between you have what globulins. Okay, we have globulins, and we have several types of globulins. You have the alpha globulins, the beta globulins, and the gamma globulins. Okay, so you have the alpha globulins, you have the beta globulins, and what the gamma globulin. We'll talk about them quite extensively today. Okay, so please follow Kindi, take down some notes, jot them down, and then make sure you try to understand most of the things now. Okay, and so we say we have albumin, we have globulins, and we have what fibrinogen. So albumin is the most abundant, and what fibrinogen is the least abundant. Okay, in between you have globulins, and you have alpha globulins, beta globulins, and then the gamma globulins. Okay, and from the material given to you earlier, you see that you have the alpha 1 trypsinogen, alpha 1 antitrypsin, and you have thyroid binding globulin. So those are examples of each of those subsections. So we'll take time and look at them extensively. Okay, and remember that these plasma proteins are present in the blood, isn't it? So we have methods of separating them. So we have several methods to separate plasma proteins. We have the plasma ultracentrifugation, where the plasma is centrifuged at various speeds, at different speeds, and then that we would have. This will lead to sedimentation of the various plasma at different rates. So when they are sedimented, they can be now separated based on what the ultracentrifugation, okay, at very high speed. So you can also have chemical separation where the plasma proteins can be separated based on what on the chemicals. Okay, maybe using precipitation, where you can precipitate by with ammonium sulfate. So saturating the plasma fluid with ammonium sulfate will precipitate what? Will precipitate albumin, and a half saturation will precipitate what? Globulins. So those are ways by which you can separate that. So you also have fractional precipitation, where you precipitate the various plasma proteins at low temperature, and also by varying the pH of the plasma. Okay, and then you add certain salt and alcohol. Okay, so you could have exposed the plasma to low temperature, passing it, and then what you vary the pH, and then you add certain salts and alcohol. Okay, and so you could also have electrical separation or electric separation, where we use electrophoresis, where you pass the plasma through a constant uh, electric current, and so the plasma will be separated. Remember, this builds on the concept or the understanding we have that proteins are amphoteric, that they can ionize as acids or as bases, isn't it? And since they contain the carboxylic acid group and the amino groups, so they can be able to ionize it as acids or bases. And then we also, this also capitalizes on the basis of the fact that what Each protein is a neutral at a specific pH. Each protein at this point, it ionizes as a base in solutions that are acidic, and it ionizes as an acid in solutions that are basic. So the principle of electrophoresis capitalizes on these properties of what of proteins. Okay, so that is actually, it was not my focus per se, but then that was just to introduce you to methods of separation of plasma proteins. Okay, but quite important what we look at this morning is the sources of plasma proteins. What are the sources? Okay, and before we look at the sources, we look at the synthesis of plasma proteins. Okay, look at the synthesis of plasma proteins. And I mentioned to you that the groups of plasma proteins, you have what? You have albumin, you have globulins, and you have what? Fibrinogen. Okay. So as almost like a rule, all plasma proteins are synthesized in the liver except gamma globulins. Okay. All plasma proteins are synthesized in the liver except gamma globulins. And where are gamma globulins synthesized? They are synthesized in lymphoid tissues by the plasma cells. Take note of this. I will ask you in one way or the other. All plasma proteins are synthesized in the liver except gamma globulins. Okay? And gamma globulins are synthesized in the lymphoid tissues by what? By the plasma cells. Okay? So we talk about what is the origin of plasma proteins. Remember, we said these plasma proteins are simple proteins or modified proteins. We have glycoproteins, lipoproteins, and conjugated proteins, all form what? Plasma proteins. Okay. And so, what are the sources of plasma proteins? So, plasma proteins are primarily formed from food proteins, or primarily formed from food proteins, but they can also be formed from what? From tissue proteins. Okay, so they are formed from food proteins. That means the proteins that are found in food, we can use them to synthesize. The body uses them to synthesize plasma proteins, and then we also have proteins from what we call tissue proteins. Okay, these are proteins that have already been stored in the tissues. Okay, so but why should we use? Why should we use food proteins? Number one is that food proteins most often are structurally similar to the plasma proteins, and the food proteins are of high biological value. That means, for example, they contain essential amino acids. Okay, and and we know that for example, we have albumin, which is formed mostly from muscle, and we have globulin that's formed from plant proteins. Okay, so we have albumin from mostly muscles, and then globulins from plant proteins. Okay, and then you also have tissue proteins. Okay, these tissue proteins could either be fixed proteins or could be reserved proteins. Okay, these fixed tissue proteins cannot be converted to plasma proteins, while the reserved tissue protein, can be converted to plasma proteins. Okay, so you have plasma proteins being formed from food proteins and from what? And from tissue proteins. Okay, and can uh, tissue proteins, we have fixed tissue proteins and have what? The reserved tissue proteins. These fixed tissue proteins can be converted to plasma proteins, and the reserved can be converted. Okay, and the reserved is also mentioned with what the dispensable reserved proteins and the labile reserved proteins. Okay, we have the dispensable reserved proteins and what? The labile reserved proteins. Okay, and the dispensable reserve proteins are mobilized from the tissues to the liver during periods of starvation. Okay, and here they are used for what? For energy production, as well as for what? As for plasma protein formation. But for labile reserve proteins, they are directly mobilized from the tissues into the bloodstream because they are structurally similar to what? To plasma proteins. Okay, and now the first part. Um, was just a regurgitation of the material we have. So now please keenly pay attention to what we'll be discussing now. Somebody's asking me what is the source of albumin. I said all plasma proteins are synthesized from the liver except gamma globulins. All plasma proteins are synthesized in the liver except what? Gamma globulins. Okay. So we would be looking at the various types of plasma proteins. And please, just to correct, I'm seeing somebody. Is it whatever Ngwagomba? I didn't say albumin is made from muscle. Okay, I said we could get um, proteins from we could get plasma proteins from food proteins. Okay, and so there are preferences. Okay, so in synthesizing albumin, because this is synthesized in the liver, and so proteins from muscle are used to produce albumin in the liver. I didn't say albumin is synthesized from the muscle. Somebody's asking how do we classify immunoglobulins? Please, let's take it sequentially. We're still talking about plasma proteins. Okay, and gamma globulins, I said all plasma proteins are made from the liver except gamma globulins, and these gamma globulins are what produces what these are immunoglobulins, and we have 5 types of immunoglobulins, and their classification is different, and we'll talk about them. GAMDE. Okay, so we'll talk about them when we come to white blood cells. Okay, and so kindly repeat on food protein and tissue protein. What do you want me to repeat from food protein and tissue protein? I'll be able to do that, but to say I should repeat it. So what do you want to understand from food protein and tissue protein? We said plasma proteins are brought in from food proteins and tissue proteins. And so these food proteins, these are the proteins in food. And these tissue proteins, we said these tissue proteins could be fixed tissue proteins or what reserved tissue proteins. Okay, reserved tissue proteins. And the reserved tissue proteins could be the dispensable or the labile tissue proteins. Okay, the dispensable, these are mobilized from the tissues during starvation, prolonged starvation, and are ferried to the liver where they are used in producing plasma proteins, while the labile are directly mobilized to the bloodstream because they are structurally similar to the plasma proteins. Okay. Okay. So are we good on that aspect? Okay. I think I've been able to address some of the things, right? Or it's unfortunate for those who are having issues with network, but then be persistent and try to see how to work about that. Okay. And so the first plasma protein we'll look at is the prealbumin. Okay, the prealbumin. The prealbumin, okay, also known as transthyretin. Okay, it's known as what? Transthyretin. Okay, the prealbumin or the transthyretin. Okay, and this is a carrier protein that is made mainly where in the liver. Remember, I mentioned all plasma proteins are produced in the liver except gamma globulins, which are produced in lymphoid tissues by plasma cells. Okay, so we have this. So we are talking about albumin or transthyretin. So we said it's made mainly where in the liver, right? And so it can also be made in the brain, specifically in the choroid plexus. Okay, it can also be made in the brain, specifically where in the choroid plexus. Okay, specifically in the choroid plexus, and it's also made in the retinal pigment epithelium in the eye. Okay, it is also made in the retinal pigment epithelium in the eye. So what are the sites of formation of of um, of prealbumin? We say it's majorly where in the liver, but it can also be made in the brain, in the choroid plexus in the brain, and then it can also be made in the retinal pigment epithelium in the eye. Okay, so what are the sites? The sites are um, the liver, the brain, and where? The eye. Okay, in the brain is in the choroid plexus, in the eye is in the retinal pigment epithelium. Okay, and what is the function of transthyretin? What is the function of transthyretin? It functions to bind. It binds T4, which is the main association with retinol binding protein. So it binds. It binds to retinol binding protein, and this retinol binding protein helps to carry what? Retinol. Okay. So what are the functions? It functions to. It binds with thyroxine, which is T4. This is a major hormone produced in by the thyroid gland. The thyroid gland produces three types of, at least several hormones. We have the T3, the T4, the reverse T3, and it also produces calcitonin. Okay, so the transthyretin binds to T4. Okay, it binds to T4, and it also binds to retinol binding protein. Okay, and when it binds to retinol binding protein, this complex now binds to retinol. Retinol is also called vitamin A. Okay, so it serves in binding. So it's a binding protein for T4 and what? And vitamin A. Okay, remember, we can find prealbumin normally in serum, which is the non-clotting part of blood, in the cerebrospinal fluid, and what? In the fluid in the eyes. Okay, so where can we find retinol? Where can we find prealbumin? In the serum, that's the non-clotting portion of blood, and then we can also find it in the CSF, the cerebrospinal fluid, and then also in the eyes. And what is the half-life of prealbumin? The half-life of prealbumin is 2 days. Okay, and we'll look at that, the importance of that. Okay. Okay, somebody's asking. I should kindly type. So call it what? The choroid plexus. Okay. Then in the eyes is in the retinal pigment. It's a retinal pigment epithelium. In the eye. So we've talked about prealbumin. It's formed majorly in the liver. It's also formed from the choroid plexus in the brain, and then we've also talked about in the retinal pigment epithelium of the eye. Okay, and we say its function is binding of T4, that's tyrosine or tetraiodotyrosine, tetraiodotyrosine. Okay, and then we have it also binds to retinol binding protein, whose association helps in binding or carrying retinol. And I said another name for retinol is vitamin A. Okay. So we said in the half-life of prealbumin is 2 days. Okay. What is the meaning of a half-life? Okay. There's a time taken for the concentration of a substance to reduce to half its initial value. Okay. So there's a time taken for the concentration of a substance to reduce to half its initial value. Okay. So we say the half-life is 2 days. So of what importance is it? Okay. So the short half-life of prealbumin makes it extremely sensitive to changes in the protein energy status of the body. Okay. It makes it extremely sensitive to the changes in the protein energy status of the body. Okay. So if you want to assess the protein energy status prealbumin is the most valuable indicator, not even albumin. Albumin is the half-life is about 20 days, okay? So the half-life of prealbumin is about two days. So that when it is produced today, within 2 days, the concentration in blood is it's, it goes to half, okay? It goes to half, okay? But what is the importance, okay? I said the importance of this short half-life makes prealbumin extremely sensitive to changes in the protein energy status of the body. Okay, it makes it sensitive to the protein energy status of the body. Okay, the importance of prealbumin in terms of energy requirements is because of the liver. Okay, the main once it is synthesized, metabolized, and excreted. Okay, so now what do we learn from this short half-life? There are two things to bring about. That will not have accumulation of prealbumin cannot occur, both in the blood, and then the fluctuations in prealbumin can be detected quickly. If the half-life is 2 days, that means you can quickly detect what the fluctuations of prealbumin. Okay, so what is the use of prealbumin clinically? Okay, so it is what is used to assess acute malnutrition and nutritional deficiencies. So we use prealbumin to assess what acute malnutrition and what and nutritional deficiency. Okay, it can also be used to predict outcomes of malnutrition. Okay, probably maybe um, uh, in the hospital, we can be able to use prealbumin as an indicator of protein energy status. Okay, in monitoring the progression of malnutrition, and probably maybe if the person is under some regimen, we could also look at the progression. Okay, so we can use prealbumin to assess acute malnutrition and nutritional deficiency. Okay, and we could also use to predict outcomes of malnutrition. So if, by measuring the blood levels of prealbumin, you can be able to look at the progression, okay, of that and predict its outcome. Okay, so prealbumin levels in the CSF have been found to be lower in patients with some neurological or neurobiological disorders, for example, schizophrenia. So we could also be you could also use to monitor, okay, or as a marker, okay, certain neurobiological disorders, for example, schizophrenia, okay, because the we mentioned earlier on that prealbumin is synthesized in the choroid plexus, okay, and since it is synthesized in the choroid plexus, it can be used as an immunohistochemical marker for choroid plexus papillomas, okay, as well as carcinomas, okay. So we can use um, it can be used as an immunohistochemical marker for choroid plexus papillomas, okay. These are what is a papilloma? It's a benign epithelial tumor, okay, growing outwardly. It's a benign, it's a mild epithelial tumor, okay, it's a tumor of the epithelium, okay. So we can also be able to use that as a immunohistochemical marker. Of course, when you prepare the choroid plexus histologically, you can be able to look at that, use that as a marker for certain types. Okay, so that is that about prealbumin. And if there are any things which we need to look at, you can build that up from your PowerPoints. And we say the lower levels are found in liver disease and nephrotic syndrome. We'll be able to look at that shortly as it comes to albumin as well. And then acute phase inflammatory response. We'll talk about that shortly. And then malnutrition. Remember, I've mentioned that it can be able to we can use it to assess, okay, the protein energy status, and we can also be able to use it to assess acute malnutrition, okay. and it's a few persons are indicating that the network is bad, but then please, we just have to make do with that. Okay, it's also seemingly not well here. So we from the PowerPoint, I mentioned something about acute phase inflammatory response, right? Okay, acute phase inflammatory response. Um, it will be good for me to educate you a little bit about acute phase proteins. Okay, acute phase proteins, and these are classes of proteins whose plasma concentrations increase or decrease in response to inflammation. Okay, so what are acute phase proteins? Are classes of proteins whose plasma concentrations increase or decrease in response to inflammation. When they increase, we call them positive acute phase proteins. When they decrease, we call them what? Negative acute phase proteins. So these are proteins whose plasma concentrations increase or decrease in response to inflammation. When they increase, we say what? They are positive acute phase proteins. When they decrease, we say what? They are negative acute phase proteins. Okay? So this response. Okay, so please kindly follow this, and then and so the acute phase reaction is characteristically involves fever. Okay, involves what? Involves fever. Proteins, not protons, proteins. We are talking about plasma proteins, so it's talking about proteins. Okay, so the acute phase proteins, and we said that what these are proteins whose plasma concentrations increase or decrease in response to what? To inflammation. Okay, in response to what? Inflammation. Okay, and when they increase, we say what these are called what? Positive acute phase proteins, and when they decrease, they are called what? Negative phase acute proteins. Okay, and the reaction is the acute phase reaction, or what? The acute phase response. Okay. Um, okay, so we have, and we say the acute phase reaction characteristically involves fever. Listen, characteristically involves fever, acceleration of peripheral leukocytes, okay, circulating neutrophils, and what? And their precursors. Okay, so we have acute phase reaction involves fever, acceleration of peripheral leukocytes, that's white blood cells, and circulating neutrophils. Okay, so what happens when we have an injury? When there is an injury, of course, and this injury can be in different, could be a burn, could be a trauma, could be an infection. In response to injury, when there's an injury, there is a response, and there's a tissue response or a body response to this injury, and this involves local inflammatory cells. Now, there are also what we call local inflammatory cells. And what are these? We have neutrophils, you have macrophages. Okay, so local inflammatory cells secrete a number of chemicals. These chemicals we call them what? Cytokines. Okay, so these cytokines are released into the bloodstream, and most notable of these cytokines are interleukin 1 and interleukin 6, as well as tumor necrosis factor alpha. Okay, so the liver now responds by producing acute phase reactants. Okay, by producing what? Acute phase reactants. What are we talking about? We're talking about acute phase proteins. And we said in response to an injury, and this injury could be a burn, could be a trauma, could be an infection. In response to an injury, local inflammatory cells, and examples of them are neutrophils, macrophages. Okay, what do? They secrete a number of chemicals, and these chemicals are called cytokines. Okay, so they secrete a number of cytokines into the bloodstream. Examples or notable of these cytokines are interleukin 1, interleukin 6, and tumor necrosis factor alpha. Okay, these are what? These are pro-inflammatory cytokines. That means they favor inflammation. Okay, so but then what does the liver do? The liver now produce acute phase reactants. Okay, at the same time, while the number of some reactants are increasing, some are decreasing. That's why we have positive acute phase proteins and what? Negative acute phase proteins. These are proteins whose concentration in the plasma either increase or decrease in response to what? In response to an inflammation. And what am I explaining at this moment? I'm explaining the fact that in response to an injury, local inflammatory cells, examples are neutrophils and macrophages, secrete, synthesize, and produce what? Cytokines. Notable of these cytokines are interleukin 1, interleukin 6, and tumor necrosis factor. And why these inflammatory cells are producing these pro-inflammatory chemicals that promote inflammation, the liver at the same time is producing acute phase reactants, okay? And at the same time, the production of this of a number of proteins is reduced, as why some are what? Some are what? Increased, okay? So in the acute phase proteins from liver can also contribute to what we call sepsis, okay? So far we are trying to discuss about acute phase proteins, okay? And so we've talked about positive acute phase proteins and what? And negative acute phase proteins, okay? So the positive acute phase proteins, they serve different physiological functions within the immune system, okay? They are part of the innate immune system. What do we say? What do we mean by innate? They are part of the natural immune system. The immune system you are born with, it is inherent, it is natural, okay? And we'll talk about it in detail when we come to white blood cells, okay? So these positive acute phase proteins are part of the innate immune system. They help to fight or ward off foreign organisms, as well as what? Responding to what? To inflammation or to inflammatory assaults, okay? So we have acute phase proteins and we have the macrophages, and they synthesize and produce cytokines into the bloodstream, notably interleukin 1, interleukin 2, that's interleukin 1, interleukin 6, tumor necrosis factor alpha, okay? Okay. And so while they are producing this and sending them into the bloodstream, these are pro-inflammatory cytokines, these are pro-inflammatory chemicals, they promote inflammation. The liver, at the same time, produces acute phase reactants, okay? So we said we have positive acute phase. Why the liver is producing acute phase reactants, or their production is increased, some of them are being what? Suppressed, okay? So for positive acute phase reactants, we said they serve as they are part of the innate immune system and have different physiological functions, okay? Some of them act to destroy or inhibit the growth of microorganisms. What are the physiological functions of positive acute phase proteins? Some of them act to inhibit or destroy the growth of what? Microorganisms. An example of these kinds of positive acute phase proteins is what we have C-reactive protein. We have C-reactive protein. You have mannose binding protein. You have complement factors. Complement factors, which include we'll see that when we come to white blood cells. You have ferritin. You have ferritin. You have ceruloplasmin. You have haptoglobin. So these are examples of positive phase acute proteins, which are inherent in the innate immune system, and they help in what? In the warding off microorganisms, as I've given examples, okay? Others give negative feedback. So others give negative feedback, okay? Others give negative feedback on the inflammatory response. For example, you have alpha-2 macroglobulin. alpha 2 macroglobulin, alpha 2 macroglobulin. You have coagulation factors. Coagulation factors. And these coagulation factors actually affect coagulation, okay? They stimulate coagulation. And by stimulating coagulation, they help to trap or to limit the infection, okay? They may limit infection by trapping pathogens in the local blood clots, okay? So we said they inhibit growth of microorganisms, and we've listed examples, and I've also said that we said they are also involved in a negative feedback on the inflammatory response, and we've given examples alpha 2 macroglobulin coagulation factors. And so these coagulation factors stimulate, actually, they are procoagulants, they stimulate coagulation, and they can limit infection by trapping the pathogen in what? In the local blood clots, okay? Also, some products of the coagulation system can contribute into the innate immune system by their ability to increase what? Vascular permeability. So they act as chemotactic agents or what? Phagocytic for phagocytic cells. So we've mentioned these are examples of positive acute phase proteins. They inhibit growth of microbes, and they also uh, give negative feedbacks. And I've given examples. And why talking about the coagulation factors or by trapping the pathogens within the local blood clot. And they can also contribute to the innate immune system by their ability to increase vascular permeability. So in this case now, they act as okay, or chemotactic agents for the phagocytes. So by what are chemoattractants? These are chemicals that attract the phagocytes to move towards the site of inflammation, the site of injury, by a process called chemotaxis. Okay, we'll look at that in details when we come to white blood cells. Okay, so those are positive acute phase proteins. So we have negative acute phase proteins. These are proteins whose concentration in blood decrease in case of inflammation. Okay, so what are examples of negative acute phase proteins? We have albumin, we have transferrin, you have retinol binding protein, you have transcortin, you have antithrombin. So these are negative acute phase proteins. Please follow the lecture and be keen and don't rely on some faith of uh, giving the video later on. Follow the lecture, please. And so we have albumin, you have transferrin, you have retinol binding protein, you have transthyretin, we have antitrypsin, you have what? Transcortin. Okay. So the decrease in these these negative acute phase proteins, the decrease in these proteins could be used as markers of inflammation. Okay. So the physiological role of the decreased synthesis of these proteins is generally to save amino acids. So when they save amino acids, these amino acids can be used to produce what? Acute phase proteins more efficiently. Remember, the acute phase proteins are the proteins we need to be able to combat the infection. So the decreased synthesis of these proteins that constitute the negative acute phase proteins is to save amino acids to be used to produce what? The positive acute phase proteins. Okay. So what is the clinical significance of these acute phase proteins? especially the C-reactive protein. It's a useful marker of inflammation. Okay, it's a useful marker of inflammation, and it also correlates with what? With the erythrocyte sedimentation rate. Okay, but then this one is used indirectly. But then when we come to ESR, we'll look at it in details, please, so that we don't mention many things here. Somebody's asking, I didn't catch what you explained about the above mentioned. So I don't even know the above mentioned. Which of the above? If it's about my post of transcortin and antithrombin and albumin, those are negative acute phase proteins. Okay, those are negative acute phase proteins. Okay, so far so good. Let me see what I gave you on the PowerPoint. We're looking at what? Okay, I picked out a point on acute phase inflammatory response, and I was building up on that. Okay, and so please kindly look at that again. So the next thing, so the first we looked at prealbumin. So the next thing we'll look at now is what albumin itself. This is an important plasma protein which you need to know. We say it's the most abundant plasma protein, and it is synthesized in the liver, okay, as a pre-proalbumin, but secreted as what? As albumin, of course. So if you will do protein synthesis, or when you come to biochemistry, they'll tell you the formation of the production of the synthesis of proteins and how this is modified, how proteins are called post-translational modifications or modifications of protein synthesis. Okay, and so it said trans-tyrosine is also a part of the negative phase acute proteins. Okay, and so I was trying to give generally the clinical significance, and so I said C-reactive proteins could be used as a marker, a useful marker of inflammation. Okay, it's a useful marker of inflammation because it also correlates with erythrocyte sedimentation rate. And I said when we come to erythrocyte sedimentation, it's the main function of it maintains oncotic pressure. Okay, it maintains oncotic pressure. Okay, which is the osmotic pressure exerted by the plasma protein that pulls water into the circulatory system, pulling water from the interstitial space, pulls water into the circulation. Okay, it pulls water into the circulation. Okay, so it exerts oncotic pressure, and albumin accounts for about 80% of the pressure of the osmotic pressure due to plasma proteins. Okay, it contributes about what 80% of the plasma oncotic pressure, and so it also helps in the distribution of fluid in and outside the cells, as well as the fluid volume, isn't it? We talked about that in our first lecture. Okay, and albumin also, besides maintaining oncotic pressure, is also important as a what? It's a non-specific carrier of hormones, calcium, free fatty acid, drugs. Please listen to this keenly. Okay. It's also a non-specific carrier of what? Of hormones, calcium, free fatty acids, and drugs. Okay. And drugs. Okay. And so it's also useful in the treatment of liver disease, hemorrhage, and shock. Okay, for example, in how do you use albumin to treat hemorrhage and shock? During hemorrhage, there's loss of body fluids and loss of ECF volume. So administering albumin, it exerts an oncotic pressure, so it helps in what? Pulling fluid into the ECF, right? Into the increasing the blood, the plasma volume. Okay, so so albumin can also be picked up by pinocytosis into the cells and hydrolyzed, and the amino acids are used to produce to further synthesize more albumin. Okay. Now, when I teach about this area, I always there's this aspect of hypoalbuminemia. Okay, hypoalbuminemia. What would be the causes of hypoalbuminemia? What is hypoalbuminemia? Is the reduction of albumin levels in blood. Okay, it's the decrease in the plasma albumin levels. So what could cause this? Well, number one thing is what we could have what decreased albumin synthesis from the liver in case of liver diseases. Okay, so we could have in case of liver disease, we could have what a decrease in synthesis of albumin. We could also have situations of malnutrition when you are not eating adequate food that is rich in proteins. Remember, we mentioned that proteins can be formed from what? Food proteins, isn't it? And we said albumin is formed from what? Muscle. So if you're not eating the right proteins or the huge quantities or relevant or adequate amounts of proteins, you may develop hypoalbuminemia. Okay, so then we've also talked about malabsorption syndrome. You could be eating, but then you have an issue with absorbing the proteins you've taken. We'll call it what? Malabsorption syndrome. We could also have what we call enterocutaneous fistula, where you have loss of contents of the intestines through the skin because there is a connection between the skin and the intestine, enterocutaneous fistula. You could also have nephrotic in instance of nephrotic syndrome. Okay, nephrotic syndrome where you have loss of albumin through the kidneys. Probably maybe you have nephritis, inflammation of the nephrons or the glomeruli, and this weaken and open the spaces between the cells, leading to what? Filtration of the albumin into the urine. Okay, so you could have nephrotic syndrome. We talked about burns, because when there's also burns, during you have burns, there is loss of fluid together with plasma proteins into outside. Okay, you loss of them through the skin. Okay, so those are ways by which those are the things that could cause um, hypoalbuminemia. We have liver diseases, you have malnutrition, we have malabsorption syndrome, we have nephrotic syndrome. You could also have even instance of even diarrhea, okay, loss of proteins in the urine. We call it protein losing enteropathy, okay? Protein losing enteropathy, you lose proteins in stool, okay? So, but what are the liver disease or this decrease in albumin? What happens? There's a loss of a key component that exerts oncotic pressure in the plasma. So, and what does albumin does? It pulls fluids into the circulation. So if we lose albumin, the fluid will stay in the tissue. So you have edema. Okay, so one of the things is what we have edema. Somebody is asking what is the loss of proteins in the stool. This is what we call protein losing enteropathy. Okay, protein losing enteropathy. So due to the loss of of um, or a decrease in albumin in the blood, so you could have there's a decrease in the oncotic pressure is lowered, and since the oncotic pressure is lowered, fluid would be trapped in the what in the interstitial spaces, and fluid would also move into the interstitial spaces. Okay, and you can develop what edema. Excuse me. We also mentioned that albumin is a non-specific carrier of hormones, of free fatty acids, of calcium, and of what of drugs. So if we don't have albumin to carry these drugs, what happens? The blood levels of the drug will be high. So we risk having what? Toxicity. So one of them is what? Drug toxicity. Okay, we have drug toxicity. Okay. Now you also have a reduced protein bound calcium. Okay, we have protein bound calcium. We have a reduced because the calcium is bound to the protein. And so we have a reduced protein bound, but then it may not affect the ionized plasma calcium. Okay, but when we come to endocrine, we'll talk about that in detail. Okay, so we've had, so we also have a possibility that, okay, because of even this lack of oncotic pressure, or this decrease oncotic pressure function of um, of albumin. It could also influence the maintenance of arterial blood pressure. Remember that what it is abundant, and so it can be it can reduce the. It has an impact on the generation of peripheral resistance, which is important in um, in setting up and maintaining arterial blood pressure. And there is this, is it Joseph Kumwenda? Please, when you ask a question, be the courtesy of politeness should be there. Okay, albumin non-specific carrier of what? No, please. And then says maybe it's another student who has followed the answer or answer. That cautiousness of in conversation is quite important. Okay, so it's great, and so we could, but then we may not have instances where we clinical conditions in which we have hyperalbuminemia. And in such a case, if we have hyperalbuminemia, can most often, as is known, should be as a result of what? Dehydration. Okay, dehydration. Okay. Then so far we've talked about that. So we continue, and so we would look at the next plasma protein we'll look at is alpha-1 antitrypsin. It's an important plasma protein. So I could ask you to any of the plasma proteins, right? Okay. Yeah. So I ask you to write the show notes on any of the plasma proteins. Okay. So alpha-1 antitrypsin is it's a protein that belongs to the serpin superfamily of proteins. Serpin superfamily. It belongs to the serpin superfamily of proteins. Yeah. Serpin superfamily of proteins. Okay. And it is encoded, it is produced or encoded in human genes of the serpina1 gene. Sorry, please. So it is it's a protein belonging to the serpin superfamily, serpina1 gene. And so it is a protease inhibitor. Please listen to the words I'll be using. Is inhibitor. When you hear the word protease, you're thinking of an enzyme that digests proteins. Okay, a protein, an enzyme that digests what? Protein. So, alpha-1 antitrypsin is a protease inhibitor. Okay, it inhibits what? Proteases. And what are proteases? Proteases are enzymes that digest what? Proteins. So, alpha-1 antitrypsin is an inhibitor of what? Of proteases. It inhibits this enzyme. Okay, follow me keenly. So it can also, the alpha-1 antitrypsin can also be called an alpha-1 protease inhibitor or an alpha-1 antiproteinase. Okay, alpha-1 antiproteinase. Protein. Okay, and actually that's a beta-1 globulin, isn't it? That's our beta-1 globulin. Alpha-1 antitrypsin is an alpha-1. Okay. Let me confirm something. Maybe if I didn't miss out something. Okay, alpha-1 antitrypsin is an alpha-1 globulin. It's an alpha-1 globulin. So we said it's a protein belonging to the serpin superfamily of proteins, and it is in humans. It's encoded in the serpina1 gene, and it's a protease inhibitor. Okay, so it inhibits enzymes that destroy proteins. Okay, so by that you can begin to imagine that it's a good thing, right? Okay, and so even though it's called antitrypsin, it doesn't only protect against trypsin. Trypsin is an enzyme produced from the exocrine portion of the pancreas and acts in the small intestines. Okay, so even though it's called antitrypsin, it doesn't just affect trypsin; it also affects other protein enzymes. Okay, so we can call it an alpha-1 antiproteinase or an alpha-1 proteinase inhibitor. Okay, an alpha-1 proteinase inhibitor. So it is a type of enzyme inhibitor. And so what does it do? It protects tissues from enzymes produced by inflammatory cells. Beautiful. Okay. Still remember when we were talking about acute phase proteins, and we mentioned that when the tissue is responding to injury, this injury this causes the inflammatory cells like neutrophils and macrophages to produce certain chemicals. Okay. Okay, so these chemicals can now act on tissues. Now, these neutrophils, besides producing the interleukin-1, interleukin-6, and tumor necrosis factor, also produce an enzyme called elastase. Okay, the neutrophils produce an enzyme called what? Elastase. Okay, when you see from the last word ase means an enzyme, produce an enzyme called what? Elastase. And what does elastase do? Elastase break down what? Elastin fibers. Okay, and where do we have elastin fibers? You could have elastin fibers in the lung tissues or in the lung parenchyma. You have elastic fibers there. When there is an inflammation, when there is an inflammatory response, neutrophils will produce elastase. And what will this elastase do? This elastase would break down, will degrade the elasticity of the lungs, leading to what? To serious, which may result in respiratory malfunctions. For example, what obtains in chronic obstructive pulmonary disease. Okay, this is what obtains in certain cases of chronic obstructive pulmonary disease. Okay, for example, in emphysema, this is what happens in emphysema, where there could be a trigger of inflammatory reactions, and the neutrophils and macrophages will release elastase, and this elastase will attack the elastic fibers of the lungs, making the lung to be fibrosed, to lose its elasticity and become hard. The same thing in smoking. People who smoke, certain chemicals from the smoke would trigger inflammatory response that would bring about what? Production of this elastase. Okay, and so what happens? This chemicals, this neutrophil elastase is inhibited by what? Alpha-1 antitrypsin. But what will happen? You have issues of what? Functionally defective alpha-1 antitrypsin, or where the amounts of alpha-1 antitrypsin are low, this elastase will act unchecked, and this can bring about what? Chronic obstructive pulmonary disorders. So what happens? The alpha-1 antitrypsin, it leaves the liver, goes into the systemic circulation, and then what? Binds to inhibits elastase. What did I say? I say elastase is produced from where? From neutrophils. Okay, this neutrophil elastase breaks down the elastic fibers or the elastin of what? Of the lungs, isn't it? And when it breaks down the elastin of the lungs, the lung loses its elasticity and becomes hardened and fibrosed, and which may result into what? Chronic obstructive pulmonary disorder. But then physiologically, the activity of elastase is inhibited by what? By alpha-1 antitrypsin, and this is produced from the liver, right? So this alpha-1 antitrypsin inhibits elastase. But what if we have an issue of a functional defect where there is what? There is a defective alpha-1 antitrypsin, or there's a deficiency? Okay, what happens? The elastase will act unchecked, isn't it? And this will bring about the conditions we've mentioned. Okay. Now, the elastic with this alpha one antitrypsin produced in the liver. What if it is prevented from reaching the circulation, from reaching the systemic circulation? It will be trapped within the liver. Also cause what? Liver cirrhosis, and this occurs in infants as well as what in adults. Okay, so it can be trapped within the liver, and it would build up in the liver and result in what? Liver cirrhosis. Okay. So in addition to belonging to the neutrophil elastase released by the inflammatory cells, alpha-1 antitrypsin also binds to elastase in localized cells, for example, in the bone marrow. Okay, okay. So where is alpha-1 antitrypsin produced? It's produced where? It is produced in the liver. And there's a point here which I want you to note. Besides liver cells, alpha-1 antitrypsin is produced in bone marrow by lymphocytic and monocytic cells. Okay, so alpha-1 antitrypsin is also produced where? By lymphocytic and what? Monocytic cells in the lymphoid tissues. Okay, so one of the things that is important about this is also helps in the motility of locomotion, in the locomotion of lymphocytes and monocytes. Okay, of course, in response to an infection, they can respond, can help in the locomotion. But even at that too, it also may also help in a localized response to what? To an infection. Okay, so this is what is important. Okay, and alpha-1 antitrypsin has been used as medication in the management of in condition individuals who have an deficiency in alpha-1. So any questions so far, we can we move on. Okay, so the next protein which we are looking at, let me follow from my PowerPoint. See the next plasma protein which we look at, looked at albumin, we looked at alpha-1 antitrypsin, and then we are looking at alpha fetoprotein. Okay, somebody is asking, so please kindly repeat on the organs producing alpha-1 antitrypsin. So please kindly explain why smokers have reduced alpha-1. Now, of course, let me start with the other part of bone marrow by lymphocytic and monocytic cells. Okay. Emphysema. How does it happen? Because these smoke, chemicals within the smoke, the network is shaking. You might not still get this. Okay, the chemicals involved within the smoke, they provoke the release of chemicals from neutrophils and macrophages, especially the elastase, and secondly, they can also inhibit the alpha-1 antitrypsin. So the elastase acts unchecked, and so it breaks down the elastic or the elastin fibers within the lungs. Okay, so this is what will predispose to that. Okay, now for individuals who are defective, who are deficient, who have functionally defective alpha-1 antitrypsin, there are synthetic alpha-1 antitrypsin which can be used and given to these individuals, of course. So preventing them from developing the pathologies we've mentioned, or predisposing them to these pathologies, especially in the liver and in the lungs. Okay. Then from the PowerPoint I gave to you, I also said the consequence of deficiency will have neonatal jaundice with evidence of cholestasis, that means stagnation of bile within the biliary tract. Okay, it could also lead to childhood liver cirrhosis, which we've mentioned that the defective alpha-1 antitrypsin can fail to migrate, to leave the site, and so will now help and predispose the individual to liver cirrhosis, liver fibrosis. Okay. Then we also mentioned about pulmonary emphysema in young adults. Okay. Now somebody's asking me to further explain what I mentioned about locomotion. Okay. Now, one of the things we said, inactivation of the alpha-1 antitrypsin by enzymes other than elastase, okay, due to inflammation and infection, causes migration of T cells to halt precisely at the site where the pathologic insult exists. Okay, so we mentioned that the alpha-1 antitrypsin is inhibited or prevented by the alpha-1 antitrypsin, isn't it? And then it would so it inhibits it and tries to cause the T cells, okay, to stop exactly at where you have the pathologic insult, okay, where you had the inflammation. Remember, these elastase products are produced due to what? Inflammation or infection. So by the alpha-1 antitrypsin inhibiting the the elastase, or it also tries to limit and to migrate, or to cause the T cells to migrate and halt at the site of the infection. Okay, and not just that, it also helps the T cells to perform a kind of immunovigilance. Okay, it tries to stay watch. Okay, they stay watch. That's the word we can use, to stay watching immunologically the tissues where it is present. Okay. Then we alpha fetoprotein, that is the protein which we need to talk about. Alpha fetoprotein. Okay. Alpha fetoprotein. Alpha fetoprotein, or what we call fetal alpha globulin, it's produced in the fetus, specifically in the yolk sac, and then also it is synthesized in the liver. So the alpha fetoprotein, or the fetal alpha globulin, is produced in the yolk sac, as well as in the liver of the fetus. Okay, and it has been mentioned that it serves the function of albumin postnatally. That means the AFP test, the alpha fetoprotein test. Okay, the alpha fetoprotein test from the maternal blood, or from the fetal amniotic fluid during pregnancy, within the 16th to the 19th week of pregnancy. So where did we say alpha fetoprotein is produced? It's produced in the yolk sac, and then in the fetal liver. And it functions almost, it performs the same role, similar roles as as um, as albumin will do postnatally. Okay, and then I was just trying to introduce the test called the alpha fetoprotein test, which is performed in women, in pregnant women during pregnancy, and the alpha fetoprotein can be assayed from the maternal blood, or from the amniotic fluid within the 16 to 19 weeks of pregnancy. This alpha fetoprotein is also expressed in the adult in some liver cancers. Okay, and this alpha fetoprotein is produced from a multigenic family encoding substances like albumin, alpha albumin, and vitamin D binding protein. So this alpha fetoprotein is produced from a multigenic family, a gene that produces other substances, and examples of them are albumin, alpha albumin, and vitamin D binding protein. Okay. So what would be the indication of low levels of alpha fetoprotein? Okay. Low levels of alpha fetoprotein normally occur in the blood of a pregnant woman. You don't expect to see that. Okay. So when you have abnormal amounts of this protein in the maternal blood, it will indicate genetic or developmental defects in the fetus. Okay, so we have low levels of alpha fetoprotein in maternal blood. So when you begin to have high levels or have levels present there, you begin to think of what? These are indicative of genetic or developmental problems in the fetus. Okay, high levels may be indicative of what? Neural tube defects. Neural tube defects. For example, spina bifida, neural tube defects, spina bifida, spina bifida, or anencephaly. Okay. Then when we have neural tube defect, this allows the alpha fetoprotein to leak through into the amniotic fluid. Okay, to leak through into the what? Into the amniotic fluid. It may also be an indicator if we have alpha fetoprotein in the maternal blood. It may also be an indication of ventral wall defects. Okay, and abnormal placenta structure. Okay, so normally we've mentioned that the levels in maternal blood are low. So when you begin to find high levels in maternal blood, they are indicative of certain things. Okay, could be neural tube defects, could be that's genetic or developmental problems in the fetus, neural tube defects. Okay, and then could also be indicative of what? Ventral wall defects. Okay, and abnormal placenta structure. We could also the alpha fetoprotein test could also be done in non-pregnancy in conditions of liver diseases. Okay. So I think this is what we had mentioned. Okay. I also mentioned earlier from the notes I gave to you that it may also protect the fetus from immunologic attack by the mother. Okay. And we said the serum alpha fetoprotein has been used as a tumor marker in adults for liver cancer. And the alpha fetoprotein, combined with other markers, has been a part of second trimester serum screening of trisomy 21. Okay, trisomy 21 syndrome. So this test, okay, and other markers, the alpha fetoprotein and other markers are a group of tests done in the second trimester to screen individuals for trisomy 21 syndrome. Okay, trisomy 21 syndrome. That is okay. That's Down syndrome. Down syndrome. Okay, and we've mentioned a couple of things. So most of them are within your within the PowerPoint I sent to you. Okay. So the next plasma protein, we'll look at the ceruloplasmin. Ceruloplasmin. It's synthesized in the liver. Another is ceruloplasmin is a ferro-oxidase enzyme. So ceruloplasmin is an enzyme. Okay. And it's a major copper. It's a major copper carrying protein in the blood, and it plays a role in iron metabolism. Okay, it is a copper carrying protein in the blood, and it plays a role in what? In iron metabolism. Okay. Okay. So it plays a role in iron metabolism. And it was first described in 1948, and it has a structural homology with hephaestin, and it participates in iron and copper metabolism. Okay, so somebody saying, can you repeat on the physiological importance in adults? I don't know which importance for which plasma protein you're talking of. In adults, it's used as a marker for liver tumors or liver cancer. That's alpha fetoprotein. Okay. So celluloplasmin, as I mentioned, is an enzyme that is synthesized in the liver, containing 6 atoms of copper in a structure, and it carries more than 95% of the total copper in healthy human plasma. Okay, so it carries what? More than 95% of the total copper in healthy human plasma. So what carries the rest? Macroglobulins. Okay, macroglobulins carry the remaining copper. Okay, and then celluloplasmin. It's as we have mentioned, it's important in iron metabolism. So it assists in the transport of iron in the plasma in association with transferrin, okay, which carry iron only in the ferric state, okay. So transferrin carries iron in the +3 state, while celluloplasmin exhibits a copper-dependent oxidase activity, which is associated with possible oxidation of iron 2 to iron 3. So it helps in the transportation of iron in a way. It helps in iron metabolism, okay. And so it binds to free hemoglobin to form complexes that are metabolized by the reticuloendothelial system, okay? By the reticuloendothelial system. We'll talk about this system later, okay? But then, what is the clinical significance of celluloplasmin? If we have a deficiency of the celluloplasmin, we will have copper will be accumulated in the liver and in the brain, okay? And this is one of the diseases we call Wilson's disease. Okay, this will lead to a condition called Wilson's disease. Okay. Okay. In the brain, it may lead to issues of cerebral ataxia, progressive dementia, extrapyramidal signs. We'll come to that probably maybe later on when we talk about Wilson's disease, especially in the basal ganglia. And then in the liver, it could cause liver cirrhosis. Okay. Somebody is just listening and asking for 5 minutes break. I'm talking. I've been talking for the past 1 hour, 30 minutes. Okay. But in any case, please, we'll soon finish. We can't afford to keep this. We are behind in trying to explain a lot. We've stayed behind. Okay. So we look at beta-2 microglobulin, beta-2 microglobulin, and it is also known as B2M, and it's a component of the major histocompatibility complex. So, yeah. Okay, we'll look at this in association with certain proteins to regulate expression of hepcidin in the liver. Okay, it functions together with other proteins to regulate expression of a protein called hepcidin in the liver, and this hepcidin targets the ion transporter ferroportin in the cytoplasmic membranes of enterocytes and macrophages for the degradation, resulting in increased ion uptake from food and decreased ion release from recycled red blood cells. Listen, please. So we said, in addition to being a component of the major histocompatibility complex, it associates with certain proteins, together regulating the expression of hepcidin hepcidin in the liver, which targets the iron transporter ferroportin, which targets the transporter ferroportin in the cytoplasmic membrane of enterocytes and macrophages for degrading, resulting in increased uptake of iron from food, as well as decreased iron release from recycled red blood cells. Okay, leading to what? Yeah. So when there is a loss of this function, it causes iron excess and what we call hemochromatosis. So there will be more iron probably being stored in the tissues, right? Okay. And so if you look at the material I gave to you, it said it's a component of the human leukocyte antigen, which is actually part of the major histocompatibility complex. Is present on the surface of lymphocytes and most nucleated cells. And it is important for a couple of things in okay, so hepcidin targets the iron transporter ferroportin, which is located on the cytoplasmic membrane of enterocytes and macrophages, responsible for degradation, resulting in increased iron uptake. Okay. Then you have haptoglobin. Haptoglobin is a protein in the humans which is important in binds free hemoglobin. It binds free hemoglobin, and compared to hemopexin, which binds free heme. Hemopexin, which binds free heme, when haptoglobin binds free hemoglobin. Okay. So when the haptoglobin binds to the hemoglobin, they form a complex that is removed by the reticular endothelial system. We'll talk about that when we come to red blood cells. Okay, and so please, you can be able to, so that we don't extend. This is at 132, so that we don't go beyond that. You will read the remaining. We have just about 4 slides, and the others are just functions of general functions of plasma proteins. Okay, you look at that. And then please, you also try to, haptoglobin is an alpha globulin. Yes, it's an alpha globulin. Okay, so you also try to look and take this subheading, these plasma proteins, and try to read further about them. Okay, and so that is what we would be able to do, and I will be able to send you details on how to submit your assignment. Okay. So please, somebody's asking if there's any book recommended for plasma proteins. In this era of e-learning, is he asking about that? You can go online and check. I may not be able, I'm not able to, I can't recommend anything yet. Okay. Okay. So thank you, please. And we will meet again on Tuesday. And on Tuesday, we'll start red blood cells. And it's going to be exciting. It's going to be very exciting. Okay. Someone is asking if they should add references to the assignment. No, not for now. No, please just read. And, yeah, maybe at most 1 or 2 references. 1 for the first assignment, 1 for the second assignment. So it should be a relevant reference. Okay. So you submit via the means, I'll ask the class reps to communicate with you. and I may be able to just a few documents if for others who want to read the material on plasma proteins. I got some material, so I may arrange it and send it to the class reps to share with you. Knowledge is to be shared, as the class rep said. So this is with my way of sharing. But when the time comes to pay, please you do the payment. Okay, please. Okay, thank you.